Hello dear students. Today we are having day 18, video 10 and for class 12. Today we will study about our next topic, that is microsporogenesis. Before we proceed, let us revise the previous one. In the previous video, we studied about the structure of microsporangium. And in case of structure of microsporangium, each microsporangium is surrounded by 4 wall layers. From outside to inside, from outside we are first having epidermis, then endothecium, third was middle layers, and then tapetum. From outside to inside, the first 3 layers, epidermis, endothecium and middle layers, they help in protection of the microspores as well as in the help in the dehiscence of the anther. And the last layer was the tapetum, which provide nourishment to the pollen grains, maturing pollen grains. There are some more function of tapetum. Tapetum helps in the storage of reserve food, which will provide nourishment to the pollen grains. 2 more important functions. One, they will help in the formation of sporopollenin. Sporopollenin is that material which cannot be degraded by any chemical or any agent. This pollenin will form the outermost covering of pollen grain. This we'll study later. Next is the pollen kit. This tapetum also helps in the providing the pollen kit to every pollen. It's a yellowish oily layer surrounding each pollen grain, which provide a specific odor to a pollen and helps in insect pollination. Fine. So pollen kit is what? It's a yellowish sticky layer surrounding the pollen, which helps in providing a specific odor, smell to the pollen, so that insects can attract towards that pollen and helps in its pollination. Fine. So this is about tapetum. One more thing, as all the microspores are formed inside the microsporangium, so such kind of development is known as eusporangiate. Fine. So now we have to study about microsporogenesis. Microsporogenesis is the process of formation of microspores from pollen mother cells. Let us go behind. That's microsporangium. Each Dithecus anther bears 4 microsporangia, and in case of microsporangia, they are having compact cells of homogeneous mass of cells is present over there called as sporogenous tissue, and every cell of sporogenous tissue will behave like myocyte, which will help in the formation of microspores. They are called as PMC, the pollen mother cells. Every single cell of sporogenous tissue behave as pollen mother cell, and these cells are diploid in nature, having double set of chromosomes. They will undergo meiotic division. Meiotic division is a kind of reduction division which will give rise to the formation of 4 microspores in a single time. Fine. Every microsporangium is having sporogenous tissue, and the sporogenous tissue, every cell of sporogenous tissue act as pollen mother cell or myocyte mother cell, which will undergo single meiotic division to form microspore tetrads. Means 4 microspores are produced in a single time. If we consider whether we are having tetrad of dicot or a monocot, such kind of arrangements are observed during microspore formation. So in case of dicot, we will get tetrahedral manner arranged our tetrads. And in case of monocots, we will be having such kind of isobilateral arrangement of microspores. Fine. These two are more important. You have to focus on monocots and dicots. So in case of dicots, tetrahedral arrangement, 3 are visible from the face view and 1 is present at the back of it. And in case of monocots, 4 are visible from the surface. Fine. So this is all about microsporogenesis. So microsporogenesis क्या है? It is the process of formation of microspores from pollen mother cell. When pollen mother cell undergo meiotic division, it will form 4 microspores in the form of microspore tetrad. Fine. That microspore tetrads are in this manner. In case of dicots, they are having tetrahedral arrangement, and in case of monocots, they are having isobilateral arrangement. Okay, that's all for today. In our next video, we will study about the later processes which will give rise to the formation of mature pollen grains. Thank you.